Hey everybody, and welcome to chapter number 2. We're going to be talking about the nervous system, and specifically we're going to be talking about neurons in the brain and how the brain communicates with itself. Let's get into it. Now, some of this should probably be review for you, not just from Psych 101, but probably review from your other psychology classes. If you took sensation and perception, or especially if you took behavioral neuroscience, a lot of the stuff is going to be familiar to you, so don't feel, like if this does feel a little bit redundant, that's okay. It's mostly because I want to make sure that we're all on the same page, because I'm kind of considering, some people are coming into this class straight out of Psych 101. Some of you have maybe taken some of the more kind of basic process psychology courses. So if you are new to this information about how the neuron works, how it communicates with itself, or with other neurons, feel free to pause this and follow your curiosity online. You can find tons of resources about all of these subjects. So please don't feel like this video is going to cover all of it exhaustively, because you could make not just a whole class out of what we're going to be talking about today. You can make an entire major out of what we're talking about today. And in fact, University of Amherst, right down the road, has a major for neuroscience. You can major in that, I believe. It is its own field of study. So some of the stuff might feel a little bit surface level. But it's because I want to make sure that we're all on the same page as we look to the next chapter, which is about perception. So knowing the key areas of the brain is going to be important for that reason. So we're going to talk about the key areas of the brain, the lobes of the brain, what they all do, how they interact, and then we'll talk about how we know that information. So we'll be looking at neuroimaging techniques, lesion studies, to understand how we know what's going on in the brain, and we'll talk about which of those methods might be best for the research questions that you have. So let's just get into it. The videos that I have for today, I don't feel like either of them are like mandatory viewing. I think they're both really interesting because they are case studies of the brain gone bad, essentially. Both of these gentlemen were involved in accidents where they experienced severe head trauma, which resulted in essentially brain damage. For this first guy, he's got Capgras delusion. Capgras delusion is a very rare syndrome where essentially you recognize your family and friends as a bunch of imposters, and you can't shake the feeling that they're all imposters. And the reason why that happens is because the area of the brain that is responsible for recognizing people and recognizing people, that are close to you, that area can no longer communicate with the area responsible for processing emotions. And the way that this is usually talked about or discussed is that the area like the hippocampus, the parahippocampal gyrus, and even the fusiform face area can no longer communicate with the amygdala. And so you have all this recognition of who these people are, but none of that kind of emotional attachment, and that leads people to think that they are imposters. I'm not spoiling the video because there's still a lot more information where that came from. I will warn though that it is a little bit of an old video, so it does kind of look like somebody recorded it from a VHS off of a Game Boy Color watching PBS. And then we're going to talk a lot about Phineas Gage because it's a psychology class and we have to talk about Phineas Gage. It's in my contract to talk about Phineas Gage. Let's get into it. So cognitive neuroscience, I talked all last chapter about how new cognitive psychology was, and it's already kind of, and I mentioned it's kind of branched off into other areas now, and one of those really key popular areas is cognitive neuroscience, which is really just cognitive psychology with neuroimaging techniques, if I'm being honest with you. And the way that usually works is that you have a bunch of psychologists that are running an MRI machine or a PET machine or an EEG, and trying to interpret the differences in brain activation that they get as a response to stimuli that were presented to people. So it's essentially like using neuroimaging to look inside the black box. If you remember that from chapter 1, the black box problem is using neuroimaging to kind of look inside the black box to help us understand how input is transformed into the output of our behaviors. So usually what this means, and we'll talk a lot about this in this chapter, usually what this means is that I'm going to use MRI, fMRI, for example. I'm going to say brain activity is measured by fMRI specifically because MRI is just structural. It's just looking at the structure of the brain. fMRI, the F stands for functional, and that's where you start looking at changes that occur in the brain. So I'm going to use that because it's probably the most popular of those techniques in the literature. So usually we'll have people lay down in a scanner, and then you have them do some kind of behavioral task. So maybe you show them an image with a face and then an image without a face, and you're interested in what area of the brain is responsible for face perception, something we'll talk about in chapter 4. And whenever you show somebody an image of a face, right, you have them look at that, you can compare that image of the brain, or that recorded activity of the brain. You can compare that with whenever they're looking at an image of something else. And then you can look at all the activation that causes. You subtract those, and what you're left with is the areas unique to just looking at faces. Not just looking at anything, but just looking at faces. So I know that this is kind of complicated. We're gonna unpack those ideas in just a moment, but that's kind of a preview of what we're talking about. So we've talked about this as input and output, right? So input is kind of like the stimuli of the environment that you encounter. Maybe it's what you're reading, maybe it's what you're hearing. And then we have the output, which is going to be our behavior, so our response to that stimuli. And so the question is, well, what's happening in between? And this is where we start talking about stuff in terms of information, that the information from the environment is transcoded into or transduced, if you want to talk about this with perceptual language, that is transduced into a neural signal, and that neural signal is represented by action potentials and neurotransmission. So essentially you have the stimuli that is kind of perceived and then converted into a signal that our brain can understand. And then from there, it's going to go to different parts of our brain. It's going to communicate with itself for a little bit, and then we have some kind of behavior as a result of that. I was going to make sure that I touched on all of these things, and I did. So how do we know this? We know this through years and years and years of not just autopsies and, people taking out microscopes to look at brain tissue, to look at specifically neurons and things like that. But we also know this because of using neuroimaging techniques, using something that we call the subtraction method to understand what these big networks do. Okay, so let's now kind of shift gears a little bit and talk about the neuron. Okay, so the neuron is the building block of the nervous system. It is kind of like, it's, I was going to say it's like a cell, but it is a cell. A neuron is a cell that essentially can communicate with other cells around it. And so this is a pretty typical neuron. I say typical. It's a very simple neuron. This is an extremely simplified version of a neuron. We'll look at some pictures of some in just a moment. You can see how crazy complicated and detailed they are. But generally speaking, you're going to have neurotransmitters floating around, as you can see here that I'm drawing. These little chemicals that we know as dopamine and serotonin, acetylcholine, GABA, norepinephrine, I already said GABA, epinephrine. So all of these neurotransmitters that are floating in the synapse, they're eventually going to make their way to these dendrites. These dendrites are these branch-like structures that are going to be on the lookout for incoming neurotransmitters. Once these dendrites get enough of those neurotransmitters, essentially enough for this cell to quote-unquote know that the next cell, the cell behind it, is communicating with it, that it will then participate in something we call integration. Integration is whenever the cell essentially decides to fire. And I say decides with quotes around it because it's not actually thinking, right? It's a simple kind of biological reaction. You get enough neurotransmitters to latch onto these dendrites, and eventually different channels are going to open up in the cell body. And what happens is a flood of sodium and then of potassium flow in and out of this, and that does that very quickly. And I always remember sodium and potassium. That sodium is positive in this case, potassium is negative in this case, these ions are rushing in and out of the cell, and that causes an electrical reaction. So this cell body, which is what you see right here, sometimes it is called the soma, that once this cell integrates and it creates this electrical potential, that electrical signal is going to flow down the length of the neuron. So now, once we have this sudden rush of ions in the cell, it is going to now shoot that electrical signal down the length of what's called the axon, which is this like long tunnel that extends all the way down to other neurons. So there's going to be other neurons down over here. Below this, if you want to think about it like in that direction. So this electrical signal is then going to come down this length, and once it reaches down to these axon terminals, sometimes you'll hear them called the terminal buttons. I think terminal buttons might be an old school term. It's what I use. I feel like people these days are more likely to call it the axon terminal, so I'll try to call it the axon terminal when I can. But that electrical signal is going to then elicit a reaction out of these terminals to release neurotransmitters. So now we have neurotransmitters that are going to be floating in this gap between neurons all over again, so that they will float. They're going to latch onto the next neuron, who is then, if it gets enough neurotransmitters, is going to fire itself as well. So that is, like a crazy fast crash course of this. And I want to talk a little bit more about that electrical impulse now. So I talked about the electrical impulse that starts in the cell body and then travels down the length of the axon. That has a very specific name. It is called the action potential. The action potential is a huge deal for these cells because cells, these neurons, have two methods of communication, an electrical channel and a chemical channel. The electrical channel is within the cell. Essentially, it is the cell telling the other side of it that it should release more neurotransmitters for the next neuron in line. So this is all electrical inside the cell. Across the cell, though, these neurotransmitters are chemicals, and so because they are chemicals, they behave totally differently, right? And that's why, part of the reason why these neurotransmitters, there's lots of different kinds of neurotransmitters. I named a few earlier: dopamine, serotonin, acetylcholine, things like that. But we only have one type of action potential. The other thing I want to mention about the action potential before we move on is that the action potential, not only is there only one kind of it, there's only one strength of it. That the action potential fires at the same rate, at the same strength every time. We refer to this as the all or none principle. And basically what that means is that an action potential is like a toilet flushing. Now, once you push enough on the handle of that toilet, and once it begins the flush, it does its thing and then it's done, right? But it doesn't matter if you just barely pressed it real slow and then let off real gently. Once you reach that threshold, it's going to flush. And if you hit it really fast, it's still going to flush at the same rate, right? So it's not the strength of this electrical signal that matters. What matters is the frequency. So how quickly are those signals firing? That is ultimately how the brain communicates with itself. I'm going to talk about this a lot throughout the semester, and in my sensation perception class, I always talk about how the brain has one language, and that language is action potentials. That is how the brain communicates inside of itself. And in that language of action potentials, there are only 3 words: faster, slower, or same. And because that is the only thing that the brain can understand, because or I should say that these neurons internally can understand, because the electrical signal is the same strength every time. That really what matters is how fast is that signal. Are we getting a lot of them? So is this cell firing is it firing a lot now, or is it firing less frequently, or is it firing the same? So faster, slower, and the same. Those are the 3 words of that language. Okay, so I made a point in chapter 1 about information theory, and about how we think about the brain kind of like a computer. And this is a really great example of kind of we made this point about thinking about the brain as a computer, and then later on we found that neurons have this capacity of firing or not firing. That remember, there is no differences of strength. It either fires or it doesn't, and it matters if it's firing faster or more frequently, I should say, or if it's firing less frequently, but that it's only firing at one strength. This means that a neuron is binary, that it is either on or it is off. It is represented as a active or inactive, or to simplify that, as a 1 or as a 0. That's what we mean by it being digital or it being binary. So a strong stimulus that you encounter doesn't mean that neuron's going to fire harder, but it means that it's going to fire more often. And so computers work the exact same way, actually. That when we talk about the circuits of a computer, that we are talking about lots and lots, billions of operations that are represented as simple as 1s and 0s. When we talk about the computer language, the way that computers understand languages is binary. But just kind of an interesting kind of, we came up with this metaphor, and then we found out like, oh yeah, they do behave like computer circuits. Okay, so I guess we got kind of lucky with that metaphor in that case. And so once that impulse travels to the end of the neuron, it is then going to release neurotransmitters, and then it goes to the next neuron, who, once it gets enough neurotransmitters, then it's going to fire and go on to the next neuron, and then go on to the next neuron, and then go on to the next neuron, and so on. And this happens all over the brain. We have like 86 billion neurons. So we have a lot of neurons, and usually when we talk about this in cognitive psychology, we don't talk about it as a neuron by neuron thing. We talk about it in terms of networks of neurons. That we talk about there are different networks of the brain, and that sometimes they communicate with each other depending on the circumstances around us. And you can kind of see, I mentioned that this cell back here was very simplified. You can kind of see here that there's different kinds of neuron structures, that they all have dendrites and axon terminals, but their shapes may be a little bit different depending on where they are in the brain and what their function is. Okay, so we have 4 lobes of the brain. I'm going to go through each of these, and they're going to come up all semester, so I would recommend doing your best to try to remember where they are and what they do now instead of later. So here is my mnemonic strategy. This is something that I'm going to shout out. I'm going to shout out Dr. Kent Bodley. He's not watching this. He might, though, because I did get an awesome email from him one time. I had uploaded a video for a class, and he came across it. I don't remember how, but he emailed me and was like, hey, it's so cool that you saw that. But he's my undergrad mentor, and in chapter 1 I talk about how my undergrad mentor was more behaviorist leaning. So I'm always thinking, whenever I'm talking about anything cognitive psychology, I'm always thinking like, what would Dr. Bodley say about this? How would he react to this? Because that imaginary Dr. Bodley in my head keeps me in check, makes sure that I'm not using too much anthropomorphized language in my explanations here, or simplifying things too much, because he would always do a good job of not doing that. Anyways, so shout out to Dr. Bodley if you're watching. Hope you're doing great. Haven't seen you in forever. If you're ever up in New England, let's go get some donuts or something. But he came up with this great mnemonic strategy that I never forgot. And basically, as he was explaining the areas of the lobes of the brain like this, he said, remember this pattern: frontal, F; parietal, P; occipital, O; temporal, T. That goes in, clockwise order. So moving from the front, going around the back, and then underneath. And if you need to remember this, that you should FPOT. F-pot. The brain is a valuable thing, so reject marijuana. F-pot. That was essentially what he said, and it always stuck with me. So if you're ever having trouble remembering the lobes of the brain, just remember to F-pot, and that'll help you remember where they are, maybe. So let's talk about what each of these areas do now. So the frontal lobe, we're going to be spending a lot of time with the frontal lobe this semester because the frontal lobe is used for what a lot of people think of as kind of like the quote-unquote human or higher order behaviors and cognition that we have. So when we talk about things like planning, when we talk about reasoning, decision-making, imagination, creativity, logic, those types of things, deep organization, that all those things are happening in the frontal lobe, and usually specifically in the prefrontal cortex. That's kind of like the all-star here. So the frontal lobe is used for a lot of those types of behaviors. That's where we'll be checking in with them throughout the semester. Then let's go from F over to P now. And so when we go over to P, we got the parietal lobe, and that's going to be used for a lot of spatial processing. So kind of understanding our bearings around us, knowing where things are in relation to one another. Is also going to be used for a bunch of sensory mechanisms as well, specifically kind of like understanding where our bodies are in space. A lot of that is going to come down to the parietal lobe's behavior or functioning. Then we're going to go over to right behind that to the occipital lobe. Occipital lobe is really easy to remember because it essentially just does one thing. It is responsible for vision, and that is mostly it. Some of that is going to be maybe some early stages of recognition, but for the most part, we're just talking about visual processing for this. And so anything vision takes place in the back of the head, which is kind of strange. And if you want to know more about that, you got to take sensation and perception, where we'll talk more about it there. After that, we have the temporal lobe. The temporal lobe is going to be used for a lot of language and a lot of hearing. And so it's going to be used for the primary auditory cortex, where hearing and sound is first processed. But we're going to be checking in with it when we talk about Chapter 4 and we talk about object recognition, visual object recognition, and how we recognize faces and common objects. We recognize those in the temporal lobe. All right, so let's talk a little bit about these other subcortical structures now. I'm actually going to pause the video for just a moment and refill my coffee. I'll be right back. All right, sorry about that, but we are back. And I had to straighten out this subcortical structures because it was all messed up and the font was all weird. Anyways, so those are the four big kind of lobes, the four quadrants of the brain. There are a number of structures that we're going to be talking about throughout the semester, though. I just wanted to highlight a few of them that are beneath the surface. So where should we start? I'm going to start with the hippocampus because we got 3 whole chapters on memory that we're going to be talking about this semester. So we're going to be spending some time talking about the hippocampus. It's implicated a lot with memory, specifically memory consolidation. So it's not necessarily that memories are stored here, is that the hippocampus's job is to really distribute those memories, those events across the brain, and ensures that they are accessible. The thalamus is used for a lot of sensory regulation. In sensation perception, we talk about this a lot. The sensory gateway, I talk about it as like a bouncer at a nightclub that will let in some signals and not let in other unwanted signals. The cingulate cortex, this is sometimes specifically the anterior cingulate cortex. This is going to be used a lot when we talk about task switching, when we talk about attention, when we talk about decision making. We got to talk about the amygdala, though. The amygdala is, I already mentioned at the very beginning of this chapter, the amygdala is really small. It's almost like the shape and size of a walnut. Shape and size of an almond, I should say. And it is used for emotion. The seat of emotion is what a lot of people call it. But when we talk about feelings of disgust, feelings of fear, those are generally processed, or the amygdala is active while we experience those things. So a lot of emotional reactions that we have take place in the amygdala. So those are some of the big areas that we'll be talking about also throughout the semester. So I wanted to give you an fMRI example to talk a little bit about how we know what areas of the brain are active using neuroimaging. Okay, so we're going to say that you have an MRI scanner, and you're going to use the MRI scanner to measure fMRI, again, functional magnetic resonance imaging. So we're going to be indirectly measuring blood flow. And the idea, the principal philosophy behind this stuff is that we are indirectly measuring blood flow, which we are going to call the bold signal. You'll see that in the textbook. The bold signal means blood oxygen level dependent signature. Signature. Why did I say signature? Bold level. But the bold signal, I said signature instead of signal. Sorry, I had a little brain fart there. So it's essentially measuring how much blood is around, and the idea is that the areas of the brain that are used more have to recruit more blood to keep that area active. And so if you can track where the blood is in the brain, that can tell you what areas are active. And if that seems strange, well, you can do the exact same thing with your muscles in your body, right? So like if you are working out just your right arm, doing curls or whatever, you're going to be getting a lot of blood flow to this arm and not as much to the other arm, so you know that one has been active, and you can tell where it has been active and where it has been used. Anyways, that's the idea behind how this stuff is understood. So let's say that we wanted to try to figure out what area of the brain is responsible for hand movement. So what you would generally do is you're going to measure the brain. You're going to scan the brain. You got this going on. This person's laying down, and now we're going to tell them, all right, we want you to lie still and look ahead. Can you do that for me, Gregory? Can you lie still and look ahead? And then while they do that, they're going to measure the brain again. And so while this person is lying still and looking ahead, we see there are some areas that are active. That is not surprising. The brain is always active. Not 100% of it is active all the time, but it is always active in some capacity. And I gotta go ahead and say, like, you see it in movies and books where people will talk about how the brain is, we only use 20% of it, and imagine the possibilities if we could unlock the other 80% or whatever bullshit. It's all fake. That's not true. Stephen King, I think, wrote about that in a book. He didn't do any research for the dead zone, and now I have to explain this every semester, so thanks a lot, Stephen King. I love your books. I'm only just mostly just joking. If you are watching the Stephen King, send me an email, tdaniel@westfield.ma.edu, and let's go grab a coffee or something. Anyways, so the brain is always active even when you don't think you're doing anything. So this person, we're just going to be scanning their brain while they're not doing anything, and we're going to call this, sometimes you'll hear it called resting state signal, where somebody is not doing anything, and we're just looking at the baseline activation that is active in the brain while we are at rest. Now we're going to ask this person, okay, we want you to wiggle your hand now. And so now if you measure this, you can kind of see the areas of their brain that are active. We're going to have some overlap, so some of the stuff was already active, right? This is probably just the default baseline level of activation, but we got this new area right here. So if we wanted to, we could call it quits right now, and we could say that area is responsible for wiggling our right hand. But we're not actually done here. at least I'm too skeptical to go with that explanation, because it could be the case that this thing is active, this area might actually be for any movement in the brain, any movement on the body. So maybe this is the area of the brain that controls movement throughout the body. And if that's the case, then it's not just the one for the right hand, so I'm not super satisfied with that explanation. Okay, well, it might also be that anytime you hear something where you are going to move, this area is active, so it's anticipating movement. Maybe it's that. I don't know. There's a couple of different explanations of what this might be, so we need to find some other control condition, right? So now we're going to try another condition. We want you to wiggle your right foot now. So now this person wiggles their right foot, and now we have a different area as active. So this is encouraging. That tells me that it might be that this area that was active right here might indeed be for just the right hand, because we have another condition where somebody's following instructions, they are moving, and they're moving the right part of their body, the right hand side of their body, and it's not the same area. And so this is what we call the subtraction method, where you take activation from doing some kind of behavioral task, and you subtract from that your control conditions. Maybe it is your activation from resting, maybe it is your activation from following some other kind of instruction, but you're going to subtract all of that other signal so that with your just what's left must be the stuff unique to that specific behavior. So in other words, if we were to just scan this piece right here and not look at any of this other stuff, then I might conclude that this area right here, and right here are all responsible for moving my right hand. But as we mentioned, those areas were already active. They didn't have anything to do with moving our hand, which is why we have to subtract that activation so that we're left with just that unique area. That's the subtraction method. That is how we use neuroimaging to help us peek inside the black box to understand what's going on in the brain whenever it is communicating with itself, when it is processing information. All right, so I just mentioned all of that stuff. I'll give you one more example here. This is something that I did whenever I was in grad school. We were looking at military veterans, and we were showing them images. Some of them were violent, some of them were non-violent, and we were interested in people that had post-concussive syndrome as a consequence of military action. And whether or not that post-concussive syndrome, so that brain fog that you get after concussion, if that had any kind of difference in how people process violence. And so what we would do is we would show them a violent image, and then we would also show them just a regular neutral image, so an image of two people interacting but no violence in it. And so that way we can subtract the brain activation from those two conditions, and whatever's left must be the violence of the image, because we already controlled for the other things like looking at an image, the attention required to focus on something, obeying an instruction, all those other areas of the brain that might be active doing some of these tangential processes. You subtract those out so that you're just left with this part of the brain that you are implicating with some kind of cognitive process. All right, the primary sensory cortex is right here in the parietal lobe, and is there anything here that I want to mention specifically? Maybe not. This is one of the things we spent a whole chapter talking about this in sensation and perception, but basically we have a slice of our brain that we feel our body, our corresponding parts of our body with. And so using the techniques that I just talked about in the last slide, this is part of how we know that, for example, this part of the brain right here is responsible for your feeling in your fingertips, and that the area right next to it is responsible for feeling the palm of your hand, and the area right next to that is for feeling your wrist, and then so on and so forth. That we are able to use neuroimaging to map out the anatomy of our body as it pertains on our brain, so that if we wanted to, we could take a little electrode and we could poke this person right here, if we poke them right there, then they'd probably say like, wow, it feels like you're touching my face or you're touching my lips, for example. All right, so let's talk about different neuroscience methods. So I want to start out with a cautionary tale, which is to be careful that you don't fall into New Age phrenology. If you're not familiar with phrenology, you've probably seen it before. Do I have? Yeah, I was gonna say I think I probably have a map of it on the next slide. So this is an example of a phrenology chart where in the past, people, and I mean in the late 1800s, early 1900s, people thought that you could feel the surface of the scalp, that you could rub somebody's head, and that you could get a good kind of feel for the areas of their brain and what they do. And so if you feel like a bump on the top of my head, so I got a big bump right here, and so that must mean that I am an extra firm individual because I have extra brain pushing up in this part of my skull. This is all bullshit. It is not how the brain works really like that at all. But people believed it and made a lot of money off of it and used it to like reinforce racist laws and stuff like that. Because if you've seen Django Unchained, the Quentin Tarantino movie, Leonardo DiCaprio's character talks about this in the last third of the movie, where he has a person's skull and he's feeling the skull and he's explaining how this person's skull means that they were not smart or whatever. And so this was used to essentially enact racist policies so that people could say like, hey, we know that that Italian people are not good critical thinkers because this part of their brain is recessed compared to people English British brains or skulls or whatever. It's like I said, it's bullshit. But that was kind of how we understood the brain maybe to work. This was phrenology. That's what phrenology was. Franz Gall is kind of like the big proponent of that. And every now and again, you will see people online picking up these dumbass theories and ideas. Sorry, I'm getting a potty mouth. I just get a little bit worked up talking about pseudoscience. But you'll see people on the internet who ascribe to all kind of ridiculous race science stuff, and they will talk about skull shapes and other kind of nonsense. It's an old trick, and ain't nobody buying it. So if you see this stuff in the wild, just ignore it because it's not true. But basically, this is kind of an old relic of how we understood the brain to work, and there was an ounce of truth here. And the ounce of truth here is that there are different parts of the brain that do different things. So the brain isn't just one organ that does everything all the time in every single way. That the brain does have different units, right? We talked about the parietal lobe, we talked about the occipital lobe, we talked about the temporal lobe and the frontal lobe, and we talked about how they do different things broadly, right? And we talked the hippocampus and how that does some stuff with memory. But that is kind of the limits of it. That's pretty much most of what it is. Like, we don't have a part of our brain that is compassion. We don't have a part of our brain that is religiosity. We don't have a part of our brain that is kindness or whatever. But we might have a part of our brain that is a network that prevents us from making outbursts. So it's like a response inhibitor. So it we do have areas of our brain that are response inhibitors that make sure that that we don't enact every crazy thought that we have, that we can think to ourselves like, wow, it would be really crazy if I just dumped this coffee on my head, and I don't actually have to do it because I have response inhibition and I don't need to do that. But there are some areas of the brain that are responsible for doing that, and if that area is damaged, then it's going to seem like I don't know, am not a very firm person, I guess. So when we talk about the areas of the brain and what they do, I think it's better to think about it like a bunch of complex networks that are communicating with each other. We don't have an introspection terminal. We don't have a extrovert module, or a kindness module, or an agreeableness module, or an openness to new experiences module. But we do have areas that do communicate and do have general specialties to them. They're just not that super specific like what we just saw. I already mentioned the amygdala, I already mentioned the hippocampus, so you get an idea of what I mean there. So when we talk about neuroscience methods, there's not really like one best way to scan a brain or to measure a brain. That it really depends on what you are doing. It depends on like what kind of research question you're asking and what kind of cognitive process you are asking. And so if you come across articles about this stuff, I think it's really helpful to ask yourself, why did the researchers use this method and not one of the other ones? Because sometimes that matters. Sometimes it's like, oh, I don't know if I can. These findings are a little inconclusive now that I know they use this method, because it's not really good for this kind of research. So let's take and also, if you haven't taken research methods 2, that's how a lot of research is. A lot of research is just trade-offs and like trying to understand like, well, why'd you use an fMRI for this question? Well, it's because it was available, right? And when we couldn't spend $8,000 per person to run a PET scan or whatever. All right, anyways, let's look at 2 different kinds of approaches for neuroscience methods. Let's look at lesions and neuroimaging studies. First, we'll tackle lesion studies. So a lesion is any time we talk about brain damage. So this damage can be temporary, it can be permanent, but it is some kind of damage that has happened to the brain, and this is part of the reason why all of the old school things that we know about the brain and how it works are from lesion studies. Before we had neuroimaging, our understanding of the brain was basically somebody would have an accident, somebody would have a stroke or something like that, and then after they passed away, they did the autopsy and they saw like, oh, this part of the brain was missing. That's why that person couldn't speak, is because that, the lingual gyrus or whatever was missing from their head. So we must know then that this area is responsible for language, because they didn't have that behavior after their stroke, and that's where they had their stroke, and etc. It can also be a product of surgical removal, as we'll see a couple of examples of that, where sometimes people will get parts of their brain removed, and that can have consequences for behaviors. So the interesting thing and the helpful thing about these studies, and I say helpful, a lot of the stuff comes at great cost, right? Because we're talking about people's well-being and livelihoods, where they get into the accidents, and then their behavior or their capacities are changed in some way, and so it does have a great cost. But in terms of its value for research, it tells us very cleanly before and after what that area of the brain might be up to. So let's talk about Phineas Gage. Phineas Gage, and I wrote about him in the textbook. Phineas Gage is this. He was a railroad worker, and I was looking to see if I had any other kind of another slide about him, but I don't, I guess. But Phineas Gage was a railroad worker, and he. Long story short, this post that he is carrying, it moved through his face, like you see here. So this is a recreation of the injury that he had based on his autopsy. But he had this railroad spike move through his entire head, clean all the way through, and it destroyed a good part of his frontal cortex up here. And so he went to the doctor. He survived this. As you can see, he's hanging out, holding that. But his behavior afterwards was changed, where people, as the story goes, that he had lots of problems kind of like with rude outbursts, and that he was less patient, and that he wasn't as good a father figure or husband, and that he would have problems drinking and stuff like that, and couldn't hold a steady job afterwards. And so people concluded that, well, it must be because this area of the brain was responsible for some of that. It must have been responsible for his ability to hold a good job and to not, drink every afternoon at the pub and to not have violent outbursts. That maybe that this prefrontal cortex helps us manage that stuff. And that's true, that we now know that it does do a lot of that stuff. But the story's a little bit more complicated than that, because whenever you look at some of his symptoms, it's hard to know how much of that was because of parts of his brain were destroyed and how much of it was because of something like post-traumatic stress disorder, because obviously this would come with a great deal of trauma, and some of the issues that he was having line up kind of nicely with some post-stress symptomologies. So the video that I linked to at the very beginning of these lecture slides kind of has a story that kind of goes into a little bit more detail in case you're interested. That's Phineas Gage. Okay, so not all lesions need to be permanent. We do have the ability to temporarily turn off parts of the brain. And this is a device called transcranial magnetic stimulation, also known as TMS. And TMS is like this wand that you place over somebody's scalp, and it administers an extremely powerful magnetic pulse, and that disrupts brain activation. And so it essentially renders that area of the brain inactive. There's a video here. It's a little bit of an old video. I wish that I could find a better video. So if you do find a good video of TMS that's better, clearer, more up to date, let me know. Send it my way, because a lot of the TMS, well, I'm getting a little bit ahead of myself, but a lot of the TMS videos that you're going to find on YouTube now are specifically about for treating depression and other kinds of psychopathologies, and that's really interesting. It's really helpful. It seems like it makes a huge difference for people, where they can come in and get some depression treatment, for example, by having this really strong magnetic pulse disrupt areas of the brain, so that they don't have to do like electroconvulsive therapy, which has a lot of stigma attached to it, for treatments of depression. But anyways, so TMS, you can take this wand, you place it over somebody's head, and if you were to like place it over the area of the brain that's responsible for someone's hand movement, and you were to use that wand, then they would not be able to reach out and hold something. They would not be able to grab something. Their hand would be temporarily active. And you can do this throughout all the different areas of the brain. In this video, you're going to see where somebody has simple problems simply like taking a sip of coffee or even talking, that they can render impossible with TMS. There is an example of it where you can see this person's getting their brain zapped. All right, another example of a lesion study are split brain patients. This is a fascinating series of studies that were done from Sperling and Gazzaniga. Sperling and Gazzaniga go on to be extremely influential neuropsychologists. And Gazzaniga also would write some extremely influential Psych 101 textbooks, very widely used books. So it may have been the book that you used in Psych 101. So if you saw it had a really long G name with a Z in it, may have been Michael Gazzaniga. But basically, these researchers were interested in these patients who had their corpus callosum severed. So the corpus callosum is the area of the brain that is a neural bridge. It's a series of neural fibers that connect the two hemispheres, the left hemisphere and the right hemisphere. And a treatment for severe epilepsy during this time was to sever that, because if you have a seizure starting in one hemisphere, then that way it won't cross over, it won't transfer over and take over the other hemisphere. So it's extremely valuable in that way. And it didn't seem to have, super serious side effects, which was surprising to everybody. That it seemed, you would think that maybe like if you had your left hemisphere and right hemisphere unable to communicate, that maybe you have two personalities, right? And that you fight yourself or something. But that's not what happened. But after some time, people did start recognizing some really strange stuff. And they started recognizing, for example, that if somebody sees something out of the right side of their visual field, that part of the visual field is processed in your left hemisphere. And what would happen is that if that, like, so let's say that you see something in your right visual field. Let's say I see a picture of a gold coin, right? And so that goes over into my left hemisphere. I could tell you that I saw a gold coin because my left hemisphere is really good at language. That a lot of folks, their speaking, their language faculties are in their left hemisphere. If I then take and show somebody something else in their left in their left visual field, that is going to go over to the right hemisphere, which doesn't really have language. So then, if I then ask you, what did you see? You would not be able to say what it was. And now maybe you got your arms crossed and you're like, yeah, that's what I would predict. That's not so interesting. But here's where it gets really spooky. That if you then give that individual in their left hand a pen and ask them to draw what they saw, they will tell you, I don't know. I can't tell you what I saw. But then you tell them, no, close your eyes and use your left hand to draw what you saw. They will draw what they saw in their left visual field right there, which goes to the right hemisphere, which handles the left hand, which is why it has to be the left hand, because that's where that information is stored. So that is an example of a split brain research study. Really fascinating stuff. Video attached down here. You can check it out. Highly recommend it because it's fascinating stuff. We don't have a whole lot of split brain patients these days because people don't do this for severe epilepsy anymore because there's other treatment methods. But if you want to see it in action, check out that video. All right, a couple of neuroimaging techniques I want to mention specifically here. We talked about the philosophy behind fMRI, the idea that what we're doing is we're looking at indirect measurements of brain activation because we're looking usually at where blood is, and that tells us what areas of the brain are active because of that. That's not true with EEG, which we'll look at in just a moment. So here are the three major techniques that we're going to be talking about this semester, so let's get into it. EEG is the most direct because it is looking at electrical activation, which means it is looking at the action potentials that are created inside of those neurons throughout the scalp or throughout the brain. And so you've probably seen this before where you have like a research patient who has on one of these cool caps like this, the little skull cap, so that, with all these electrodes on it. And what that's doing is it's measure electrical activation throughout the brain. The nice thing about it, so I mentioned all research is a trade-off. And so it's important to know, why is the researcher using this specific measurement for this specific cognitive process. EEG is really fast. It gets its signal almost instantaneously, which is really nice if you're looking for really quick, fast cognition. So if you're looking at decision making, for example, you might want to go with the EEG because a lot of decisions are made in the blink of an eye, right? So it's really fast. It's non-invasive, which means you don't have to you don't have to have somebody in an MRI scanner. You can even have somebody sleeping for this. You can have them sitting in a chair. They don't need to be in a hospital or anything like that. So it's fast, it's relatively cheap and affordable, and it's non-invasive. The downside of it, though, is that it is relatively weak. So what that means is that there's a lot of activation that's not happening on the surface, and because of that, that signal may not be able to reach the electrodes. The deep brain stuff is hard to measure. And another reason why it's hard to measure for that reason is not just because it has a weak signal, but also because that signal is very noisy. And by noisy, I mean that there is a lot of data noise. There is a lot of variability in that signal, which is hard to draw, is hard to interpret, is hard to draw conclusions from because it can be such a messy signal, messy data set for that. So it's a great tool, but not always great for everything. If you're if you're interested in studying sleep, it's the best game in town. If you're looking at measuring like eye movements, like the ability for the brain to measure eye movements, that's probably the way to go too, because that's a really fast thing. All right. PET, or positron emission tomography. This is whenever you create a radioactive isotope, and well, let me take a step back here for a second. So with positron emission tomography, what people do is that they get injected with a radioactive isotope, and then they get their brain scanned. And that's what you're seeing right here. These different kinds of activation patterns is because this person is getting scanned, and they're looking at where is that radioactive isotope in that person's bloodstream. So the radioactive isotope, they are manually created by different labs, and those isotopes are usually created with a specific function. And maybe that function might be that they are going to bind onto receptors that normally detect dopamine. So if you do that, and that's not exactly what's happening in this figure down here, but just, give me a second and I'll try to explain. If you inject somebody and they have, this is going to bind on to dopamine receptors, and you scan their brain, now you can see like, oh, these are the areas that have very rich dopamine receptors because it's deep red. The blue ones means that there's much fewer. And then the yellow is that there's, kind of a moderate amount. And then you could use that information, then compare normal individuals with people who have Alzheimer's disease to people that just have dementia. And you can use neuroimaging that way. So it's a slower type of research, because you're not really looking at behavior in the moment. You're looking at more kind of chemical changes in the brain. So the advantages is that it is the best way to look at kind of chemical reactions in the brain in a large way like this. It does measure blood flow because you are looking at where those radioactive isotopes are in the bloodstream. But the disadvantages of this is that you can only do so many trials a year, because you are injecting people with radioactive stuff, and you can't do that very much because it can cause cancer. So you can only usually do like two sessions a year for that reason. So it makes some of that stuff hard to do, that research hard to deal with. And also, like, as I mentioned, these are slow acting changes, so that blood flow takes about 30 seconds to really kind of make its change. And so it's not really all that good if you're like trying to look at attention, for example, which is something that happens very quickly. It's also extremely expensive. I mentioned about $8,000 to run a single participant for something like that, because it costs a lot of money to create that radioactive isotope. So it has its purpose. It's a very specific purpose. Yeah, PET, really, really cool stuff. I don't want to dismiss it at all, because my sweet spouse, she is a researcher who primarily does PET research, and she's awesome at it, and does a lot of really cool stuff with it. But she's also not a cognitive psychologist, so she's a clinical psychologist. Okay, so let's talk about fMRI. We talked about it a little bit already, but just to recap, this is measuring blood flow, that bold signal, the blood oxygen level dependent level. And the way, its pros and cons here. The advantages is that it's really good at getting specific, nitty-gritty, high-resolution images. So this over here, PET, in EEG, these are like standard definition. These are not really good at getting precise locations of activation. fMRI is much better at that. Back whenever I was in grad school, people were like, you have to have a resolution of about 2 millimeters, which means that you take the brain and you kind of divide it up into 2 millimeter blocks, which we call voxels. Voxels are like kind of the unit of measurement whenever you are measuring things in the scanner like this. But since then, 2 millimeters is like, oh, that's really good. That's really high resolution. it's gotten smaller than that. You can do resolution depending on how strong the magnet is. You can get it at 1 millimeter or even smaller than that, depending on how strong that magnet is. So you can get really, really precise within a millimeter of precision with these images. The temporal resolution, so in terms of how quick it is faster than PET. It's not as fast as EEG though. So we're talking about a change that it takes about 1 to, it says 4 here. I might backtrack on that a little bit. Some people will say 6, so between 1 and 6 seconds for blood to make that change there. So keep that in mind. If you're still studying attention, that can be really tough because attentional changes can happen, within milliseconds, and so that is good for EEG, not really, really good for fMRI. Another advantage is it doesn't have any radiation involved. It's relatively cheap compared to PET imaging, and it's okay if you get scanned multiple times a month with this, because there's no long-lasting outcomes from having your brain scanned with that magnet. The disadvantages for this is that because you're tracking indirectly the blood flow, sometimes it's slow and it's not super precise for that reason. And also you got to worry about ongoing brain activation. So this is why when we were using the subtraction method, that we had to subtract out that unrelated brain activation to try to find the meaningful signal there. But also, like, if you have somebody doing multiple things in the scanner, you have to make sure that some of that old activation is not overlapping with the new activation. And sometimes it can be hard to control for brain differences across participants. So which is the best cognitive neuroscience method? Well, it depends. It depends on what it is specifically that you're looking for, and that would probably be a great test question, right? So what would be best at looking at sleep? What would be best at looking at attention? What would be best for looking at short-term memory? What would be best for looking at long-term memory? Think about those critically because that'd be good to know. I'm going to go through one more neuroimaging example here. So this comes from a landmark study from O'Craven from, I think, 1999. So, and I should also say, this is going to show a little bit of my prejudices here, that neuroimaging before 1995, I feel like is a little too early for me to be super confident in. But honestly, anything before 2000, at least to me now, feels a little too early to be super conclusive about. Some of that stuff still holds up, and I think O'Craven definitely still holds up, but that's part of the reason why, there was a lot of stuff done in the 90s, a lot of important stuff, but the neuroimaging techniques have gotten so much better in the past 30 years that it really outshines a lot of those old findings now. Not that they've necessarily been reversed, but that they've been more refined. Those findings and conclusions have been more refined in the time since then. But this is a classic one, so I wanted to talk about it with you, and it's really straightforward. So basically, people came into the scanner, the fMRI scanner, and they were given an instruction. We want you to look at the face, or we want you to look at the house. And what they did is they showed participants both at once. So you see a face and a house together. And this was a really interesting kind of question to ask, because if you're thinking about what are your eyes detecting, your eyes are soaking up the entire visual field, right? So your eyes are seeing, or are detecting, I should say, your eyes are detecting a face and also detecting a house. But if I ask you to focus primarily on the face, you see the face more. And if I ask you to focus more on the house, now you see the house better. So what's going on there, right? So your eyes are your eyes are seeing both window and eye. So why is it that based on the instructions you can see one or the other more clearly? What about now, right? So same idea. So they were given instructions, look at the face, focus on the face, or focus on the house, and what they found was, and I was wrong. It was 1998 that some of the stuff was done. And actually, I see Nancy Canwisher here. We're going to talk more about her in chapter 4, so I'm glad that she came up now. She is a legend in this stuff. Nancy Canwisher, also another person for your, if you're bored and you want to know more about kind of more recent influential researchers, she is a great person to check out. She once shaved her head so that she could draw a diagram of the brain on it, and I should find that image. Or actually, you can Google it. You can find it. She's really cool. But anyways, so this is her data that we're looking at right here. But whenever people were asked to look at the faces, that the fusiform face area in their temporal lobe was active. But whenever people were instructed to look at houses, they had a different area active. They had the perihippocampal place area was active. So even though the eye was detecting both the face and the house, the reason why we were better at seeing the face or better at seeing the house, depending on the instructions that we got, is partly because of the differential activation in the brain. If we have the instruction to look more at the face, the fusiform face area is going to be more active. If we are asked to look at the house, the perihippocampal place area is going to be more active. So this is a quick example of how, using neuroimaging, we can scan the brain and then look at what did we give people. So what was the input? We can look at the processing, and then we can better understand their output. So why is it that their behavior, they said that they were better able to see the house versus the face? And again, you can kind of see it here, where here they asked people to look at the house and then look at the face. And so you can kind of see where that happens on this timeline. This dotted time is when they got the instruction to change their focus. And on this axis, you're seeing the blue line is going to be the fusiform face area activation, and the red is the perihippocampal place activation. So when they were asked to look at the house, you see that they have an increase for the perihippocampal place area, and then when they're instructed to look at the face, now the fusiform face area jumps up, and it is now responding at a stronger signal. And then we see the exact opposite whenever the instructions are reversed. So they're looking at the face, and then when they're instructed to look at the house, we see the perihippocampal place area is the more dominant activated area at that place. All right, so I think that is a this is a good place to end it. Thank you so much for tuning in. If you have any questions, feel free to shoot me an email and let me know. If you reply with a YouTube comment, I do appreciate it, but I don't usually respond to those because I'm not very good at using social media. But be sure to like and subscribe, tell your friends, tweet me, share this stuff on Instagram and on TikTok or whatever. I'm just joking. I don't really care. Thank you for tuning in. I do care about you tuning in and lasting all the way to the end. And I hope you have a awesome day. I will see you later for Chapter 3, which is about perception and cognition. Bye-bye.